we go. No more sound checks. We're going live.
So go ahead.
Hey, you want to get close to the unit here?
Yeah. Good morning. I'm Rick Oil, Director of the Center for the Study of Adolescent Risk and Resilience, or C-StARR. On behalf of my colleagues in C-StARR, it's my pleasure to introduce Scott Swartzwelder, Professor of Psychiatry and Behavioral Sciences in the School of Medicine at Duke with a secondary appointment in Psychology and Neuroscience. Scott's research focuses on the effects of alcohol on the brain in rodents and humans, particularly during the period in the lifespan when the brain is still developing. His influential work has been published in top drawer journals such as Addictive Behavior, Alcoholism, Clinical and Experimental Research, Neuropsychopharmacology, and Scientific Reports, and been cited more than 12,000 times. His work's been funded by many grants, most from NIH, and most of which he's principal investigator. We're pleased to count Scott among the Duke addiction scientists affiliated with C-StARR. Scott is co-author of 4 trade books that aim to translate for practitioners and the public at large the neuroscience literature on how the use of alcohol and other drugs, including chocolate and caffeine, affects the adolescent brain. His trade book, Buzzed, co-authored with Cindy Kuhn and Bill Wilson, and originally published in 1998, is now in its 5th edition. One Goodreads reviewer of Buzzed wrote, "Buzzed is a pretty remarkable book, designed to cut the BS out of propaganda for and against drug use. Buzzed is essentially a reference book for all psychoactive substances, legal or illegal, from caffeine to nicotine to ecstasy. It gives a breakdown of what damage is caused to the brain by excessive chocolate consumption, and it tells you why caffeine makes you feel so jumpy sometimes. It takes away a lot of the myths about some recreational illegal drugs, but also gives frightening examples of what long-term use can do to the human body and the brain." So a nice sort of synopsis of what that book is about, and a lot of what the message has been that Scott has been a part of championing. As you're about to see and hear, Scott has the uncommon ability to describe findings from bench science using language and examples that are understandable and interesting while staying true to the science. So please join me in welcoming Scott Swartzwelder.
Thank you, Rick. All right. Well, thanks very much. I really appreciate that. There have been other things written about Buzz that use slightly different language, but I'm glad you chose that one. Right. Yeah. Well, most of them have been flattering, but some of them have been a little more salty. But anyway, I really appreciate being here. It's a great pleasure to talk to you all and all of you out there in Zoom land. I hope you can see me and hear me okay. What I really like about being here today is that it gives me the opportunity to talk to a really diverse group of scientists who are interested in adolescence and risk and addiction and that whole confluence of factors that influence how we develop as human beings and how we get on a trajectory toward productive adulthood. And so for somebody like me who has spent much of my time in the very basic science world, although I am a clinician as well and I do human studies, it's just nice to be in a really multidisciplinary format. I'm looking forward to more. I want to introduce you to the folks in my lab now, because usually people do this at the end and you'll be finished hearing me, right? You won't be paying attention anymore in 45 minutes or so. So I want to point out that some of the folks in my lab and in my partner lab. Alex Marshall is actually at NC Central. He's assistant professor. He's a co-investigator on my alcohol grant now. Katie Healy, a postdoc, unbelievable postdoc with me. Kayla Wachuwu is a graduate student with Alex at Central and is part of our group. Amelia Bell is our research technician, former Duke undergraduate. And we have these wonderful 5 Duke undergraduates working with us now. Megan Rabe, Michael Greenstein, and Jenny Liu are all seniors. Michael and Megan are doing senior theses in my lab right now. And Michelle Huang is a junior, who's going to be a thesis student with us next year. And Bianca Da Silva has just started, in fact, so recently that I couldn't even get a picture of her. So, anyway, these are the folks that are working with me now. The presentation is going to be much more historical, so they didn't contribute to all of these data, but I wanted to acknowledge them because it's a great group, interesting people in the lab. Really founded a lot on Duke undergraduates, which I have always done and always have really appreciated. I also want to point out with this slide that I don't know if you all know it or not, but here at Duke, there are 3 different consortia that are focused on adolescence. In addition, to your center, there are principal investigators for the NADIA consortium. That's me, right? So NADIA is neurobiology of adolescent drinking in adulthood. So it's a basic science, molecular, cellular focused group, nationally sponsored. It does 9 or 10 labs nationwide, right? Sponsored by NIAAA, looking at the basic science of what happens when you expose individuals to alcohol during adolescence, and then they grow up into adulthood, right? The NCANDA, there is also a component of the NCANDA and the National Consortium on Alcohol and Neurodevelopment in Adolescence. That is a human studies focused version of the NADIA. So NIAAA is sponsoring these 2 national consortia that look at adolescent alcohol exposure and downstream effects in adulthood. One doing the basic science, one doing the clinical/human studies kind of research. So it's an incredible thing that we have here at Duke, that we've got components of each of those national consortia here. And then the Consortium for the Study of the American College Student, with which I'm involved, was started here by John Looney, who is a psychiatrist in the School of Medicine. And his interest is really focused on that transitional time between, when you live at home and when you're out in the world. And so study of college students. So that nexus, that transition to adulthood is the focus of that consortium. So it's a really rich environment with your center and all those foci here. So the opportunity to bring some of that together is really exciting to me. And so I appreciate you inviting me here. So in terms of the effects of alcohol on the adolescent brain, so our group here at Duke is often credited with doing the foundational work on that and showing, the early studies of how alcohol differentially affects brain function in adolescents compared to adults. And we were early in the game, that's for sure. But I also want to point out that none of that would have happened without my late friend and colleague, Linda Spear. And I don't know how many of you know or know of Linda's work, but she was really the first person to start to think about modeling adolescence in rodents, okay? Like trying to figure out what happens in the lifespan of a rodent that parallels adolescence in a human. Critical thing to do in order to try and do an animal model so we can dig into any of this cellular level stuff that we've been so productive with over the years. And Linda really laid that groundwork, studying the adolescence from an endocrine standpoint, from an early pharmacological standpoint. And she was also a good friend of mine, and she didn't let me get away with any crap. If I said something stupid, I knew about it. Linda. Kind of like our pal Cindy Kuhn, if you all have hung out with Cindy. But I don't know if Cindy's tuned in or not, but I'm sure she'll take me to task at some point for something. Anyway, so talking about the adolescent brain and alcohol effects, I always view adolescence as a time of brain building, okay? All of development is, but adolescence is a time where really the circuits in the brain are getting sculpted into their final form. Right? They're getting turned into that level of circuitry that is going to either set an individual up for success in the adult world or not, okay? And there are a lot of factors that can influence brain development during adolescence. Drug effects, obviously, we're going to talk about that some today, but nutrition is important. Sleep is important, and sleep during adolescence is different than sleep during childhood or adulthood. Exercise, both physical exercise and cognitive exercise, are important for neurodevelopment. The impacts of stress and trauma are of obvious importance, as are the social experiences that an individual has, and the things that we learn during adolescence. I'm going to talk a lot about learning because I study the hippocampus, and that's sort of a good lens through which to view adolescent brain changes and alcohol effects. This slide is from long ago, from my colleague Jay Geed up at NIMH, who back in the day, he did these nice MRI studies looking at changes in neocortical gray matter volume across early life, starting at 5 years. Basically, as this thing gets more blue and purple, you've got more of an adult type neocortex in terms of gray matter volume anyway, right? And you can see there's not much at full maturity here at 5 years of age. By the time and that's changing, through the early adolescent period, you get out here into 20, 22 years of age, which I think is where these last images are from. And you've still got some pretty good swaths of neocortex that are not in their final adult gray matter form, particularly here in frontal lobe regions and in the lateral temporal lobe regions, which are important for some of the stuff I'll be talking about later. Importantly, these regions of the brain that are undergoing development are underpinnings for a lot of cognitive and cognitive affective functions that we rely on, that people rely on in adulthood. Memory, problem solving, cognitive flexibility, the integration of affect with cognition, and planning and execution of plans. So really executive functions writ large, but also learning and memory functions in a more fundamental way, which I'll talk about. But we can't really talk much about adolescence without spending a little time on the very austere looking G. Stanley Hall, who was a psychologist early in the 20th century, who really was the first person to characterize adolescence as a developmental epoch, right? Before that, there was no real study of adolescence as a developmental period. He referred to it as a period of storm and stress, right? And talked about difficulties, how that influenced behavioral interactions with peers and family and so forth. So he was really the first one to do that, right? So if you think about this concept of adolescence from an academic standpoint, from a scholarly standpoint, it's quite new. We're talking 120 years ago at most, right? So as a culture, as a society, as a community of scientists, we're still coming to terms with what adolescence is. It's pretty new as a concept about humans. And so, a lot of work has been done, but I still all the time find that I interact with people in the lay public world, giving lectures at schools or whatever to policymakers, and people still have these odd conceptualizations about adolescence. So that tells me that this work is important, and it tells me that as a society and as a culture, we're still coming to grips with understanding what adolescence is. So this research and the research that you all are doing in terms of adolescent, resilience and risk and so forth, really critical and important for our understanding of that developmental period. So what's distinctive about adolescence? I like to say that the adolescent brain is built to learn. Okay, it's a tremendous period of synaptic plasticity in the brain during adolescence. So the stuff that you learn in adolescence, it sticks with you, and it's there. Learning is critical for success during adolescence, right? So anything that you do to promote the capacity for learning and acquisition of information during adolescence is critical for success during that period. And learning during adolescence is critical for success in adulthood, because you take it with you, okay? David Rubin has, he and I used to talk about, he has these studies where he looks at memory, how salient memories are at different phases in people's lives. And the memories that people formed in their teens and early 20s are more salient when that person is 60 than what that person remembers from when they were 50. Right? So it tells you that there's something going on during this period. I think it has to do with synaptic plasticity, right? Your receptors are open in so many ways during adolescence. So it's really an interesting period in that way. The memories and skills formed are long lasting. Of course, brain development, related to cognition, affect, reward integration, and executive functions in general are really coming online. And then, of course, differential drug sensitivity. So that's, of course, all this plasticity is a double-edged sword. On the one hand, it creates an enhanced opportunity for learning, the acquisition of behavioral repertoires that are going to help us be successful out into the future. but it also endows us with an enhanced vulnerability to the disruptive effects of some drugs. And the drugs that have been studied most with respect to adolescence have been alcohol, cannabinoids, and nicotine. And we've done a little bit of work in each of those areas. I'm going to focus on alcohol today because it's a ubiquitously used drug during adolescence, obviously, but it also, there's some features to it pharmacologically that make it a good lens through which to view brain effects. And so I'm gonna focus on alcohol. So some of the core neurobehavioral effects that alcohol has include learning, right? And learning, of course, being, I always think of learning as a building block for higher order cognitive function. If you can't learn and remember things effectively, it's hard to do much with that information, right? Sedation is another principal effect that alcohol has. That, of course, relates to consciousness, motor function, and the regulation of drinking, because when you start to have enough alcohol on board to feel sedated, that's the signal that most people are able to use to say, okay, time to stop, right? So it's a very important signal in terms of drinking regulation. And an anxiolysis, or decrease in anxiety, which of course relates to, affect, the experience of disinhibition that people feel with alcohols because of the anxiolytic effect in part. And it relates to risk taking. Because when you're not anxious, you're not scared, you sometimes do stupid things, right? So all of these things really relate to alcohol effects and are really important in adolescence. I'm going to focus a lot on learning and somewhat on sedation. I'll leave the anxiolysis for another conversation. All of these alcohol effects are differentially regulated by alcohol during adolescence, every one of them. Okay, sometimes alcohol's effects are more powerful, sometimes they're less powerful. We'll talk about that, right? But all of these three core neurobehavioral effects of alcohol are different in adolescence compared to adulthood. And so that's what I'm going to walk through now and tell you a little bit about how we know that. So let's talk about memory, right? The first one, learning and memory. So the effects of alcohol on memory range everything from very mild deficits in explicit memory being what you know, right? Or what you've learned as opposed to implicit memory, all the way out to total deficits in explicit memory, like a blackout, right? Where there's a chunk of time during which your brain just wasn't forming any memory, right? And the thing about blackouts is that, people think about them kind of as a joke. They kind of think it's funny. A lot of students say, oh yeah, like I got so drunk, I can't remember anything. And that gets integrated in our society in some ways. And this clip I'll show you kind of illustrates that. I hope you all can hear it now. Here's the test.
Winchester had the most unforgettable time of their lives. But this morning, they can't remember a thing.
So, in case you missed it, that was the trailer for the cinema classic Dude, Where's My Car? And the reason I show that is because it's a whole movie based on this idea of these guys get drunk and get blacked out, right? And how much fun they had and how weird that is, So my point is, it's not funny, right? As a society, we think of blackouts as something funny or, oh, look what happened, but it's not trivial, right? And just as an example of how it is not trivial, these are data from a study that we did some time ago. We did this at a university. I will not name the university, but let me just say that it is a prominent Southeastern institution. What we did is we went and got people who, after they had been out, drinking, and we got compatriots who had been with them. And so we looked and said, well, what happened during blackouts? And these are things that happen that people do not remember happening to them, right? But their compatriots, were able to report on. Anyway, just to walk through a couple, they, 33% insulted someone, 27% spent money they didn't intend to spend, 25% engaged in some form of sexual activity. I want to pause here right? Because for somebody to engage in sexual activity and not remember it, that's huge. We are built to remember sexual activity, right? That's what we're about. And so if you got a drug that's able to wipe those kind of memories out, that is a powerful drug, okay? 16% got involved in an argument or a fight, 16% vandalized property. I want to point out an important sex difference here, right? It's 30% of men, only 5% of women. So guys get drunk and bust things up, right? And then women don't. That's important. Had unprotected intercourse, 6%, had unwanted intercourse, 5%, right? These are serious things, right? 2.5% of those students had driven a car and did not remember having driven a car, okay? And amazingly, none of them had been arrested. But that might be a policy issue. That's not my bailiwick today. But so the point is, blackouts are serious. They're very serious. Dangerous things happen. I like to tell students, you know, in order to cause like a 2 or 3 hour gap in your memory, not using alcohol, I'd have to hit you in the head hard enough to knock you out for about 10 minutes, right? So now a head injury is totally different than an alcohol dose, right? Let's be clear about that. The point is, it's a big neurological insult when you shut the brain down enough to stop its encoding of memory, right? And people don't think about it that way, but it's valuable to do so. Okay, so why do we have blackouts, right? And why is memory disrupted so much by alcohol? I think it has to do with the structure called the hippocampus, which you're all probably familiar with, that hippocampus has these reciprocal interconnections with basically a whole rest of the brain, particularly neocortical areas, right? That are bringing in sensory information from all the modalities. There's all this back and forth connection, but everything is getting filtered down through the hippocampus. And the hippocampus is kind of keeping it available and alive so that through these reciprocal interconnections, the cortex can tap back in and say, oh, remind me what happened over there, right? And so the hippocampus is this repository of plasticity that's available to the rest of the brain, particularly neocortical areas, where ultimately those memories are going to be stored in a long-term way, right? Hippocampus doesn't do that with any piece of information forever. It's time limited, but it is critical. If you shut down hippocampus function, you just don't consolidate memory for that period of time. And we'll see some information about this shortly. So hippocampus. Now, here I want to show you a study just to illustrate the powerful effect that alcohol has on the hippocampus. This is a rat on what's called a Y maze, because it's shaped like a Y, because behavioral psychologists are very creative in the way they name things. But we have a rat on this Y maze, and we've got an electrode implanted into its hippocampus, and it's recording from a single neuron, all right, and called a place cell. And in the hippocampus, place cells are designed to fire, become active, and induce plasticity when the animal's in a specific location, particularly when they're trying to learn about that location, okay? So what's going to happen when I activate this movie is that you're going to see this animal's going to walk down into this section of the maze, and when he does, that cell's going to start firing, and you'll hear like, right? That's that cell firing action potential. So I want you to just listen to the robust firing that goes on when he hits that part of the maze. Right, a lot of firing just in that region of the maze. It's represented visually over here, the firing patterns. Right, so good, robust firing. Circuits are working in the hippocampus. Now here's an animal with a dose of alcohol on board, okay? So listen to the difference. Far less when he walked through. Now he's going to go up. He actually hangs out in this area. There's no firing. No fire a little bit more when he walks back through. Right? You can hear the marked difference in the firing rate. Now, I want to point out this guy has alcohol on board, but he was walking around just fine, right? He was not falling off the maze and staggering around. He was not slurring his little rat words, right? He was walking around, pretty much okay, but his hippocampus was markedly compromised in terms of that cell's ability to encode information and provide him with information about where he was in space so that he could learn it, right? So I think that's a good illustration of the power that alcohol can have on hippocampal function. Now, the hippocampus is always striving to maintain a delicate balance of excitation and inhibition. Somehow I've lost my curve. Oh, there it is. And you always want that balance because you don't want to tip it too far in one direction or another, right? If you do, you run into problems. For example, the two main neurotransmitters in the hippocampus that regulate this balance are glutamate on the excitatory side and GABA on the inhibitory side. If you push things too much in a direction where you increase glutamatergic function and or decrease GABAergic function, you can have hyperexcitability, even to the point of epileptogenesis or seizure generation within those circuits, okay? So you don't want too much excitation and or too little inhibition. On the other hand, if you reduce glutamatergic excitatory activity and or increase GABAergic activity too much, you can get memory deficits, okay? So you really want this thing balanced. It tips toward learning when you're trying to learn, and it tips toward, sedation when you're trying to relax, but basically you want it kind of hovering in the middle. You don't want too much of any of these things. And that's what the hippocampus is doing a lot, is just sort of navigating this balance of excitation and inhibition so that it can provide us with the capacity to encode information when it's salient and important, not when it's at random, okay? So, first question we endeavor to ask about alcohol in adolescence is learning-related brain function affected differently by alcohol in adolescence than in adulthood? So here's our hippocampus wiring diagram from before. And here's just a cutaway version of a hippocampal slice. And all this information from the neocortex is coming in. It's going through a trisynaptic circuit in the transverse hippocampus and then feeding back out into these neocortical circuits. It's a really wonderful system for promoting plasticity and keeping information available. So there's a real hippocampal slice in the chamber, and we can stimulate in one part of the circuit, record in another part of the circuit. So we're looking at this very specific signal, right? And which is subject to plasticity when learning occurs. And we can determine the effects of alcohol on the capacity of this circuit to induce plasticity, right? Well, one of the ways that circuit induces plasticity is by the activation of a particular kind of glutamate receptor called the NMDA receptor, right? You activate NMDA receptors, you induce synaptic plasticity, you promote learning. So that's what we looked at first, okay? You looked at the effects of alcohol on NMDA receptor function. And what you see here is an ascending series of alcohol concentrations that we put onto the slice. And this is the percent, this is basically the amplitude of the NMDA, the learning-related synaptic signals in that hippocampal slice. In adult animals, you get what you would expect, this nice linear offset when you put on higher and higher concentrations of alcohol. But when you do that in slices from adolescence, the shape of the curve is much different. There's a much more powerful effect. And importantly, the impact of that effect is most obvious at lower alcohol concentration, relevant to what humans take in, okay? By the time you get way out here, they start to come together, and that's a very high concentration. But down here, this is where the differences are. So this is really important. It suggests that the adolescent hippocampus is far more sensitive to the effects of alcohol on this learning-related plasticity mediating glutamatergic receptor. We also did that experiment in single neurons, which I'm not going to bother to go through. But the point is that even at concentrations as low, this is the amount of inhibition of that receptor function in adolescents compared to adults. And you can see across the dose spectrum, it's different. But I want to point out 5 millimolar, that's like a drink or two at most, right? That's a low concentration of alcohol. 10 millimolar, totally human relevant. But you see this marked difference in the potency of alcohol to inhibit that memory-related signaling in the hippocampus, even at a single neuron level, which is what these experiments represent. The next level is to look at a phenomenon called long-term potentiation, which is a manifestation of synaptic plasticity. okay? LTP is, it's not all that memory is, but it is an underpinning, a physiological underpinning of memory, which is mediated in large part by NMDA receptor. So it would make sense that if alcohol inhibited NMDA receptor mediated function more in adolescence, it would also inhibit the induction of long-term potentiation more in adolescence. So we do this experiment, same setup. We get a signal when you stimulate here and record here. You get your baseline signal. Then we stimulate that circuit in a way that mimics what happens in that hippocampus when the animal is actually learning. Okay, so we can mimic that kind of activity to promote a learning or a learning-like reactivity in that circuit. And you come back later and that signal is much bigger. Okay, same stimulus pulse, but now because of these intervening pulses, it's much bigger. That's the basic empirical definition of long-term potentiation, right? So we do this experiment in adolescent slices compared to adult slices. And so the adults here are the circles, the adolescents are the triangles, and the baseline response is the filled symbols, and the alcohol response is the open symbols. And first, you look at the adults, right? So we give the stimuli, we get this nice long-term potentiation, okay, in adults, 20, 25, 30% increase in the response. But there isn't really any effect of alcohol on that. Okay, so at this dose of alcohol, you can put alcohol in and get effects in adults, obviously. But with this particular dose, we hit a sweet spot because it didn't do anything to LTP induction in adolescents. But now look at the data from in adults, but look at the data from adolescents. First of all, they produced more LTP to begin with than adults did. Remember, the adolescent brain is built to learn, right? They're primed for plasticity. This is an example of that, okay? But when we put this dose of alcohol on and gave that stimulus, it completely knocked out the capacity to produce LTP. So it just basically limited it, right? So that's a huge effect, right? That's a really big effect. Now, across a dose response curve, you see this, like I said, this is the sweet spot for doses, right? You can put on doses that don't do anything to any age. You can put on doses that knock it out in everybody. But in this medial area, right, where we're talking about human relevant doses, it's really knocking it down in adolescents and not doing much in adults. Not going to bother with the raw data. Somebody got to tell me when I'm out of time, right? Because I don't have a clock. So if alcohol does this, it inhibits NMDA transmission more in adolescent, it inhibits the induction of LTP in adolescents, it ought to inhibit learning more as well. So we did that experiment. And for this experiment, we used it's called a Morris water maze, which some of you are probably familiar with. And this animal is in this little tub of water and is swimming around. This is a platform that he can climb up on to escape, right? But he can't see it because it's submerged just below the surface. So he has to swim around, swim around, swim around, swim around. And eventually he bumps into it and climbs up and goes, great, I'm out, right? This is good. So the platform's in a particular space in the room. And so he stands up there and then looks around for a while. And we've got things on the walls so he can learn. So it's like a big picture of Jimi Hendrix over here, a black circle there, somebody else over here, and then the animal gets put back in the water in a different place than he had put in before. So he can't just learn to swim out and swim left, right? We want him to learn a spatial task because spatial memory is very hippocampally mediated. So this task is a really good assay, if you will, for hippocampal function. Right. So we did the experiment in adults and in adolescents where animals either got no alcohol before each of their sets of trial, or they got one of two doses of alcohol. So let's look at the adults first. Here's a control condition. And what you're looking at here is the amount of distance that the animal had to swim across, like 25 trials in order to reach a memory criterion where they're pretty much swimming to the escape platform. In adults, the low dose of alcohol had no effect. When we pushed it up to a higher dose, you start to see an effect there, right? But look at the adolescents, right? Here's the baseline, here's the controls. The low dose of alcohol was much more potent at impairing the learning process, right? And a higher dose just made it worse, right? So very similar to what we saw in NMDA function and LTP, in the behavioral learning situations, it's the same thing. the animals are just much more sensitive to the effects of alcohol if they're adolescents than if they're adults. But rats are not the only animals that we're interested in. We're also interested in humans. And so we did an experiment. This was done by Sean Atchison in my lab, again, many years ago now, but worth going over. We brought folks in and we gave them alcohol and got them up to just below the illegal limit, right? 80 milligrams per deciliter is illegal to drive, right? We brought them right up below that. Then we snagged a window of time in here and did some memory testing, right? Now, I actually, this is all people from 21 to 29 years of age. I expected them all to be the same. We couldn't do adolescents because we couldn't give, or what I was thinking of as adolescent, teenagers, because you just can't give them alcohol, right? So I said, oh, well, let's just use young people and then we'll go and do it in older people later and, just see what we got. But so that's what we did. We ran them through a bunch of memory tests, including like a figural memory thing like this, where you give somebody this card and a blank card and have them copy this, right? And then you take this card away and give them another blank card and have them do that from memory, right? And then you go off for a while and you do a bunch of other tests. Then you come back and you give them another blank card and you say, remember that thing I showed you? Right, draw it again, right? And so it looks like it would be very difficult. It's not easy, but actually, you young folks would do great. I would not. But it's a good memory test. We also did semantic learning with word lists and story recall and stuff like that. So I'll show you just the data from the figural memory thing. So I thought all of these people were going to look the same, but they didn't. The data were just all mungled up. But I decided that I said, well, let's break this group out. Let's look at the young 20s and the late 20s, right? And when I did, it was shocking, right? So here's the placebo condition and here's the alcohol condition. You look at people in their late 20s, that alcohol really didn't do much to their memory, their capacity to learn that thing. You look at the younger folks, market effect. And I'm just talking 5 years younger. I'm talking early 20s compared to late 20s. Same thing on delay. This is immediate recall. This is delayed recall. Same story, right? Very interesting data. And, I think with powerful implications for what happens when young people drink. And again, we weren't even looking below age 21, right? That, to my knowledge, that still hasn't been addressed. But so to summarize a little bit, this learning related stuff, alcohol has much more potent effects at suppressing memory-related synaptic signals, the NMDA receptor function, memory-related circuit changes, or LTP, right, synaptic plasticity, spatial memory acquisition in animals, and human memory acquisition. The story's the same, all the way up the line, from the molecular level at the membrane of the neuron, through the plasticity circuit, through the behavior, and into humans. So it's really, really nice story. But there is another feature to this story, and that has to do with the sedative effects of alcohol. And in that instance, it's the exact opposite. That is, alcohol has much more powerful sedative effects in adults than it does in adolescents. And that's in part why when adolescents drink, they look like this. And when people my age drink, we look more like that. Right? And so fortunately, as a scientist, the same thing happens in rats, right? And so we're able to study it and look at these differences in sedative potency for alcohol. So we use a simple device called a tilt plane, which assesses basic motor function. And I'm not going to go into great detail, but the red symbols are the adolescents. The blue symbols are the adults, and the farther down this is pushed, the more impact the alcohol had on the animal's motor performance. And so what you see is, pretty big difference here, right? Across time after an exposure to alcohol out to 180 minutes, there's just a much more powerful motor impairment effect or sedative-like effect in the adults than in the adolescents. Okay, that's really important. We've also done this on other measures of sedation, like alcohol-induced sleep time, which Cindy was involved in back in the day, and the intoxication scores, motor intoxication scores after a dose, in addition to this one. And the story is the same, right? Much more powerful sedative effects in in adults than in adolescents. So what does this tell us? It tells us that because of these developmental differences in alcohol sensitivity, when an adolescent drinks, they are less likely to feel impaired, but more likely to be impaired from a cognitive standpoint, right? They're not getting that sedative signal. Remember, I talked about how sedation was important because it just signaled to stop drinking. Adolescents don't get it at the same dose as adults do, right? And so they keep drinking. And even with the doses on board, they are less sedated, so they can continue to behave and do stupid things, right? And so, this juxtaposition of greater sensitivity on the part of adolescents to the cognitive effects and less sensitivity to the sedative effects, it's exactly what you don't want in an inexperienced drinker, right? It's what you don't want. So, how's my time?
It's about 15 minutes in total.
15 minutes in total, which means if I talk for 15 minutes, nobody can ask me any questions. Jeez, I got to learn to speed this thing up. Okay, so I'm just going to talk about one more thing because I do want to take questions. Talk a little bit about the long-term effects. So we've talked about difference in acute potency, right? And there are profound and important differences. But at least as important is, does this mean that alcohol exposure during adolescence has long-term effects, right? And is that more potent than if the same kind of exposure occurs in adulthood, right? So are there long-term effects of adolescent alcohol exposure? And is the adolescent brain more sensitive to those kinds of effects than the adult brain is? Well, the answer is yes, by the way, right? But I'll talk a little bit about, I'll give you a couple examples of that. Here's just a list of some of the effects that people have found, not just us, but others in the Nadia. And you have these slides, so you can go back and find these, right? I'm not going to talk about them all, but there's an interesting convergence, and I'll make this point in a couple of ways, and then I'll stop. There's an interesting convergence in this literature about the ways in which alcohol changes adult function if it's given during adolescence. And that is, there's an apparent retention of adolescent typical phenotypes into adulthood. In other words, exposure to alcohol during adolescence seems to lock in certain adolescent characteristics and perpetuate them into adulthood, okay? And this is a very interesting set of findings. So it happens at the behavioral level, okay? Now, remember, I showed you this. I hope you remember this. If you're not drunk, you probably do. That alcohol is just much more potent at disrupting learning, right? In normal adolescents than it is in normal adults. Okay, but if you take adolescent and adult animals and expose them to repeated alcohol, and then let everybody grow up, and then look at the effects of alcohol, acute alcohol, on their ability to remember things. What you find, we did this in a radial arm maze test. This is the adolescents who didn't get alcohol during adolescence. Those who did. This is the adults who didn't get repeated alcohol, and those who did. And the higher up that bar goes, is the more disruptive a single dose of alcohol was. Now remember, everybody's adults now. Okay, so the only group that was markedly impaired by this single low dose of alcohol was the animals who had gotten repeated alcohol exposure during adolescence. The ones that got repeated alcohol exposure during adulthood, they were no more impaired by it than the controls. So this tells us not only that alcohol exposure during adolescence perpetuates alcohol sensitivity to memory effects into adulthood, but that it doesn't do it in adults tells us that it means the adolescent brain is distinctly sensitive to that long-term effect. The adult brain is not sensitive to that long-term effect. That's a developmental difference that's very, very important, okay? You remember this slide too, right? That adolescent rats are less sensitive to ethanol-induced motor impairments and sedation, okay? Right? Well, so are adult rats who are pre-exposed to alcohol during adolescence, right? So they're retaining their adolescent-like insensitivity to the effects of alcohol on sedation. I'll show you just how this goes. So here's an experiment. We gave 3 single challenges of alcohol and looked at their effects on motor function, as in that experiment I described. So the first, that's the first part of the experiment. And so if you do this in adults, right, you see this, right? So the adults, they're very sensitive to those sedative effects. So you give 1 dose, you come back 25 days later, still an adult, right? Still 25 days later, still an adult, still sensitive. Exactly what you would expect. If you do this in an adolescent, what happens is when they're an adolescent, they are less sensitive to the single dose of alcohol, but they grow up, they turn into adults, and they become more sensitive, just like they should. And then you get out 25 more days, they're still adults, they're still sensitive, they're adults, okay? Now we're changing the experiment a little bit. In between test 1 and test 2, we give repeated doses of alcohol, okay? And then we come back and a couple of days after that last dose, day or so after that last dose, we give the second, and then we come back and do a third. Now, if you do this in adults, all right, first dose, they've never gotten any repeated alcohol yet. They're sensitive to the sedative effects. You give them repeated alcohol, test them right after that exposure period, they're a little less sensitive than they were. You know why? Tolerance, right? They got tolerant to that sedative effect. But then 25 days later, the tolerance is gone. They just look like normal adults again, okay? But now, and this is the one that counts, this is when you do the experiment in adolescence. First dose, they're not sensitive. Given repeated doses, they're still not sensitive. You wait 25 more days until they're well out into adulthood, and they are still not sensitive to the sedative effects of alcohol. So I think that's a very powerful demonstration of the fact that animals retain these adolescent-like sensitivities in the case of memory effects and insensitivity in terms of the sedative effects. And they carry that with them into adulthood. I think that's just a very powerful thing. So I'm not going to go through any of this. I think am I like down at 5 minutes or something? I got 7 minutes. That's so good. So I'll tell you. I think let me maybe let me skip forward just because I do want to show you that that the news is not all bad. That you see that can the effects be reversed? Right. Well, we've done some experiments with 2 clinically used drugs, donepezil or Aricept, which is a cholinesterase inhibitor. It's used for early onset Alzheimer's. It's used for minimal cognitive dysfunction, and then gabapentin, which is called Neurontin. It's used as an ancillary therapy for neuropathic pains, sometimes for seizure disorders, and sometimes for bipolar disorder as an add-on adjunct treatment. But anyway, what we find is that these drugs can reverse some of these effects if given in adulthood to animals that had alcohol exposure. And so one thing we've looked at is dendritic spines, right? The spines on neurons, dendrites that are actually bringing in excitatory or inhibitory information, right? And what we find, interestingly enough, is that AIE is the adolescent alcohol exposure. AIE decreases the density of dendritic spines on hippocampal neurons. Okay, that's an important finding, right? Because it corresponds with some of what we've been talking about. But if you give the animals 5 days of donepezil exposure in adulthood, right before you sacrifice them and take the brains and look at these, it reverses that. It normalizes that. Okay, that's really that's good, right? That's hopeful. I'm not going to get into the weeds on this molecular stuff, but we know that the FMR1 gene, the Fragile X mental retardation gene, is involved with dendritic spine morphology and density, right? And so we looked at FMR1 gene expression in this same kind of preparation. What we see is that FMR1 expression is increased after adolescent alcohol exposure, increased in adulthood, which would correlate, based on what we know about its effects on spines, correlate with a decrease in spine density. And that increase in FMR1 expression was normalized by donepezil as well. We went even farther down the molecular rabbit hole and looked at epigenetic mediation of FMR1 expression. So we're looking here at histone H3K27 expression, right? Which is, you know, it's a histone that regulates the expression of a gene, you know, through relaxed regions of relaxed chromatin, right? And so when you get an increase in this expression, it just means there's more availability, essentially, of the chromatin for epigenetic modification, and that was increased on the FMR1 gene, right? And that was also reversed by donepezil. So we got this cool effect that is, you know, reversible by donepezil, and we've got potential genetic and epigenetic mechanisms that might underlie it. And so I had about 800 more slides, but I think I'll just stop here and thank you very much for having me, and I'll be happy to answer any questions you might have.
So is the message, I'm thinking about two kind of controversial issues. One is, should the drinking age stay at 21?
Yeah.
And then the history there has nothing to do with these matters, but relative to these matters. And then is really abstinence what you want in an adolescent, legality aside, or is it better if they have some experience?
Yeah. Wow, that's a great question. Great pair of questions. As far as the drinking age is concerned, if you followed the science, you would raise, not lower, the drinking age, right? Because the brain is not finished doing its developing in a human until about 24, 25 years of age. So, you know, it's like I always told my kids, don't do anything permanent until you're 25, right? Like no marriages, no tattoos, right? Just, you know, like don't do anything permanent until you're 25. So that's what the science would say. But of course, you know, as we know, science and policy don't always match up. But I think that, you know, if I got a choice between people drinking at 21 or 16, I'll take 21, you know. But I think if nothing else, these data suggest we absolutely shouldn't be talking about lowering the drinking age. I mean, unless there were some incredibly compelling social reason. Well, like, you know, the drinking age was lowered from 21 to 18 in Maryland when I was 18, but that was when Vietnam was going on, right? 18 years old. We couldn't vote either. So we were raising hell. And so, you know, so the drinking age got lowered for a while. It only got raised in the 80s because of getting attached to highway funds, right? And it's an interesting story, you guys probably all know. But so that's what I think about the drinking age thing. In terms of abstinence, you know, there are good data in the human literature that basically just say the longer you wait before you start drinking recreationally, the less likely you are to have trouble with alcohol later in your life, right? So that's clear. I mean, people who start drinking recreationally very early in their adolescence, 12, 13, 14 years old, they're very likely to run into problems later. And so basically, the longer you delay that recreational drinking onset, the more you're stacking a deck in your favor of avoiding substance use disorder, alcohol use disorder later. So abstinence, I don't know. I can't say I'm an advocate for abstinence in any respect, but I do think that the longer somebody can delay heavy drinking or repeated drinking, the better, as I think these data suggest. Yeah.
For the groups of the 21 to 24-year-olds versus 25 to 29,
Yeah.
that difference is fascinating, but I'm curious, did you ask about how much and how often they were drinking beforehand and control for that? And so we did. Great question. That's a really good question. I'm glad you asked it. Yeah, we did our best to control for tolerance. And what we did is we screened people. We didn't want people who didn't drink, and we didn't want people who drank enough that they would be notably tolerant. So we had this window, right, of drinking. And, you know, we tried to recruit Duke students, but they all drank too much, right? So we ended up with a lot of community students. You know, they saw, oh, alcohol study. Yeah, let's go, right? But they were all the tolerant ones, right? So we couldn't bring them in. But yeah, we did our best. You know, there's
we couldn't control for the age difference, right? So there's an age difference there, but of course, that's kind of the point of the study. But yeah, we tried to control for that. We brought them in, we gave everybody a standardized meal, and so we really try to, and the error bars on their BACs are pretty tight too, so we're pretty confident with it, but that's a good question. You can't control for everything, but we tried.
Yeah, actually. By the way, Kayla's in my class. I'm taking Henry Ian's class, and Kayla.
Oh, yeah?
Yeah.
What?
Yes. This is like 269.
Oh, great.
I have it. So my question is actually from having that class, and I might like launch it because I don't think that I have a good grasp of it. But under my understanding is that what is really important to learn, I think, is the discrepancy between, I guess, at least for some learning theories, between the expectation and then what the reward is. What is happening is that discrepancy is associated with dopamine activity, which then helps
Yes.
Learn, I guess, the value of
Right.
behavior. And I was wondering if that kind of applies also to alcohol use in adolescence. Is alcohol use in adolescence perhaps also more majority? Is there maybe like they don't know what to expect? There's a discrepancy.
Yeah. Yeah, that's a great question. And the answer is yes. Alcohol does produce more effect on dopamine release, in a number of studies during adolescence. So yeah, they are hyper reward sensitive. They are less punishment sensitive. And so, yes, I think that is an important backdrop against which to interpret these kinds of findings, right? But it's also represents in itself risk, right? If you're hyper reward sensitive and you're not punishment sensitive, that's, again, it's just this balance you don't want. You put alcohol in that mix, right? And now your cognition isn't working as well, and you're wide awake instead of sleepy, right? But you're primed for pleasure and you don't care about pain. Boy, oh boy, that's a nightmare waiting to happen, right? So yeah, I think it's a really important point.
I hate to end on that note, but.
We have one more question.
All right,
We have the time for it. Someone was asking about if there's research on the medications being used in humans and if there's thoughts about using it clinically.
Oh, these particular meds? No, not right now. This is early. We just published these data, a couple of years ago. Aricept has not really been used in anybody except the elderly population for the most part. So, we'd need a lot of clinical trials on that. Gabapentin has never been used for anything like this. I get emails asking that question all the time from people who are worried and say, man, I drank a lot as an adolescent and, I'm not thinking right. Should I take some gabapentin? All right. And I say, probably not. But I think what it does, whether or not either of those drugs is ever used clinically in this way, it gives us some target areas to look at, right? So cholinergic function, right? GABA mediated or thrombospondin mediated, which is really what gabapentin does, thrombospondin mediated function. So it points us in therapeutic directions, but people are not using those drugs that way now. You're welcome. All right, great. Thank you all. This was fun. All right.
So I guess